I'm basically going to be giving a little micro-history, I suppose, let's see if I've got this, of law and hormones, or I think rather law in hormones and hormones in law. So what makes a substance toxic? Well, one answer that conventional toxicology would give is that the dose makes the poison. And that relationship between higher doses of a substance and an increased physiological effect is referred to generally as potency. And in the 1940s, perhaps no substances were as resolutely positioned as potent than were so-called sex hormones. So so-called natural estrogens like estrone and estradiol and the synthetic estrogen DES were being crystallized and synthesized in researchers' labs throughout the 1920s and 30s. But how did estrogens come to be therapeutic drugs? So in 1940, menopause was not really constructed as a disorder yet, and estrogen was arguably a drug in search of a disease. There was evidence from animal studies as well throughout the 1930s that these estrogenic substances were carcinogenic. So how did sex hormones in the 1940s begin this explosive transformation into drugs that in the second half of the century would come to be prescribed to many millions of women? I mean, by the 1990s, estrogen was the most, Premarin was the most prescribed drug in the United States. So on this question, there is a really rich history of feminist scholarship in the history of science, mostly in STS, which gives a lot of rich insight to this question, and perhaps most foundational is Nellie Udshorn's 1994 study, which showed how hormonal drugs emerged, in her words, in the triangle of the laboratory, the pharmaceutical industry, and the clinic. But my research suggests that the triangle is actually a square, because regulators also form part of this network. And specifically, I'll be arguing, and I argue in my research, that the enactment of estrogenic substances was partly achieved in Canada in the 1940s through the practices of a group of regulators, which I mean broadly. So a group of regulators, including scientists, lawyers, coordinated by the Department of National Health. And following Anne-Marie Moll and her take on actor network theory, I say that these actors' regulatory practices basically performed or materialized estrogen as a multiple object. And in its potency and its multiplicity, estrogen in turn catalyzed some new regulatory techniques in Canada, and most notably a new kind of labeling. This is just an image of the nascent products that were available in the Canadian market in 1940. So the legal backdrop to this story is the Food and Drugs Act, one of Canada's oldest statutes in the 1940s. Its purpose was seen as twofold. It was basically to protect public health and to protect the public's pocketbook. So it had this anti-fraud purpose. And in the 1940s, that dual purpose was increasingly pursued by standard setting. So standards for quality of a drug, identity of a drug, purity, and potency. And this was done by the delegation of statutory powers from Parliament to the Governor and Council to Cabinet. And importantly, as a piece of context, in contrast to the United States, where the Food, Drug, and Cosmetics Act, as of 1939, under that act, companies had to prove the safety of a drug before it could be marketed, as of 1939. But in Canada, new drugs did not need regulatory approval. So Premarin and DS were actually sold in Canada, marketed here before they were approved for sale in the US. I should note some of the humans, some of the human actors. In this micro-history, the most important human actors are the members of the Canadian Committee on Pharmacopeal Standards. Now, this committee was instigated by Dr. Vellian Henderson, who was a leading prof of pharmacology at U of T, and he had long advocated for a greater Canadian role in drug standards. He wasn't the only one. Since the Treaty of Westminster in the 1930s, senior officials in the department had really been engaged in a form of nation building and bureaucracy building through drug regulation and drug standards. And this is just an image that makes me laugh of a 1949 booklet commemorating basically the compounding of the Canadian state through food and drug standards. But Professor Henderson had become very agitated in the early 1940s by changes that had been made to the British Pharmacopoeia, a big word, I will say BP. Basically, a pharmacopoeia is like a recipe book for drugs. Pharmacists would be horrified by that brief synopsis, but he became really horrified by changes that were made to accommodate wartime shortages in ingredients. And it was problematic because in Canada, under the Food and Drugs Act, the BP basically was the default standard. Absent a Canadian standard, the BP set drug standards for Canada. So this new committee was formed to basically advise on new Canadian drug standards that would be in a Canadian supplement to the BP. And there were a bunch of professors appointed, professors of medicine, pharmacology, pharmacy, some departmental officials, and importantly, the minister also appointed 2 scientists from the pharmaceutical industry, one of whom was Dr. Stanley Cook of Montreal-based Ayerst, McKenna, and Harrison. And for those of you who think about estrogen, it was Ayerst and its partners that first extracted estrogens from pregnant mare's urine in 1939, and in 1941 that began marketing conjugated estrogens under the quite unforgettable brand name Premarin, pregnant mare's urine. And for the wholly uninitiated who don't think about estrogen, I should just note that this is a substance that is used in the birth control pill. It's used in hormone replacement therapy. Hormone replacement therapy is used by some menopausal women. It's used by some trans people and some non-binary people for re-embodiment purposes. It used to be used in cosmetics in Canada. There were a series of very high-profile studies in the 1970s and again in the early 2000s that connected these drugs, these therapies' uses, especially at high dosages, to a range of cancers: endometrial cancer, breast cancer, in the case of DES, to these transgenerational vaginal cancers and reproductive anomalies. But back to the 1940s. So the new committee gathered for the first time in January 1943, and the department's hormone expert, Dr. Leonard Pugsley, introduced the department's draft sex hormones regulation, which uniquely in Canadian drug regulations at that time, at least as far as I can tell, defined sex hormone to include all products, synthetic or natural, purporting to have estrogenic properties. But how to identify and measure these purported properties? Well, Dr. Pugsley acknowledged later in a journal article in 1951 that in the 1940s, and this is pretty widely acknowledged, the department really lacked any reliable reference standards for new estrogenic drugs. A reference standard being basically a little comparator substance in a little vial, a literal physical standard to compare drugs against. And he also acknowledged that at this time in the early 1940s, it was a bit of a gold rush mentality in sex hormones, and manufacturers were expressing the potency of their products in this hodgepodge of ways, using mouse units as a measure of potency, rat units, rabbit. The reference here from the very famous professor of pharmacology at Oxford is that, kind of mockingly, it remains now for the bolder spirits to discover methods in which a lion or an elephant unit may be described. So measurement was really all over the place. And as we know, you can't manage what you can't measure. So Dr. Pugsley's draft sex hormones regulations proposed that where there were no reference standards that already existed, so no little comparator vials of substance to compare against, and where stable standards weren't being provided by the manufacturer, then the methods for expressing potency and testing potency should be in the department's discretion. And this did not sit well with the professors on the committee. They felt that Canadian regulatory standards for sex hormones needed to specify methods of bioassay. Bioassay is basically just animal testing. So they felt that basically everything, in the words of the minutes, everything needs to center around the methods of testing. And the department's concern was that test methods change. This is a changeable area of science, and if we codify them, that's going to require constant, expensive regulatory amendments. So the scientists backed down when the department suggested, don't worry, we'll informally distribute to universities our internally used bioassay methods. At the same time, in early 1943, the committee began mapping out a long list of drugs to be included in this supplement to the BP. Dr. Henderson sent a list of proposed substances that included estrone and estradiol, but the department wrote back that they seem to be at some loss as to the precise object you have in mind, because rather than seeing those two estrogens as biological substances or as chemical molecules, the department saw them as preparations, as products preformed by industry that would be controlled by the forthcoming regulations, whereas the professor, Dr. Henderson, understood them as a pure and active substance that, regardless of how they were regulated, should still be defined. Dr. Cook of Ayerst also challenged the inclusion of estrone and estradiol in the supplement, also viewing them as ready-made dosages, but eventually the committee decided to include them. Oddly, the next day, the department considered the committee considered the department's draft sex hormones regulations separately. There was some debate about the adequacy of the department's internal bioassay methods, and Dr. Cook ended up reporting back on this in the fall. As it turned out, US pharmaceutical companies had no problem with the department's estrogen test methods, but it did have real concerns with the department's gonadotrophin method, and this triggered a really vigorous debate on the committee about who should decide how to measure sex hormones potency. I won't take you through the debate, but it was closed off by the deputy minister who, interestingly, was observing only this part of the meeting, Dr. Roadhouse. And his view was that it was not for the officers of the department to take a stand in any new field until they were satisfied there was a danger. The committee returned the next day to separate from that discussion to consider this monograph for estrogen. You see the end part of estradiol and the beginning of estrone. And they agreed to standardize it, and it seemed suddenly that estrogen was about to be standardized in Canada. Or was it? So the monographs contain some ambiguities. You see at the top there circled that the method of assay, the test, was left to the department's discretion. And you see at the bottom that there is some discussion about how what would constitute, how potency would be expressed, the unit for measurement. Interestingly, no estrogenic properties or effects were indicated for estrone or estradiol, although for DES there was an effect defined, which was vaginal estrus in spayed adult female albino rats. That is what, according to this soon-to-be codified regulation, made estrogen. That is what estrogen was, something that caused vaginal estrus in overrectomized adult female albino rats. This estrogenic multiplicity was enacted yet further in the spring of 1944 when the department was finalizing its sex hormone regulations. As already acquiesced to, the regulation delegated to the department the ability to express and test potency, and Elmer Dreger, a well-known lawyer, then a solicitor at DOJ, sent back a question to the department. Was the department trying to prescribe standards of potency? The department replied that it was trying to prevent manufacturers from using unreliable test methods, but by doing so, in Dreger's opinion, the regulation would unlawfully delegate cabinet's regulatory power to bureaucrats. So faced with this wrinkle, the department innovated. What if, they asked, rather than letting us decide how potency should be expressed and measured, we instead require companies to attach a label to their products? That label can, by that label, the manufacturers can tell us the unit of potency that they're using and tell us the test method that the manufacturer had used. Dreger thought that was unobjectionable. Industry was not displeased. In fact, when they were consulted on the draft regulation, they wanted additional labeling requirements because industry wanted to advertise the potency of their products. So this turn to labeling, I argue, as a governance technique for estrogen can't be understood in modern parlance as being about the consumer's right to know. If anyone's right to know was at issue here, it was the department's right to know. They wanted to know what was materializing in industry's labs. There's Dreger's opinion. There's an example of the Premarin's IRIS effort to highlight the potency of their products. And example extracted there of just how the department's and the committee's decisions on delegating potency of their products to manufacturers, how that legal decision was really embedded into these pills. How this is an example, as I will argue, of basically congealed law in a pill. So reflecting on this, and I'll just take one more minute. Reflecting on this, a year later, a member of the committee, Dean Woods of the University of Saskatchewan, expressed anxiety about this growing lack of drug uniformity due to this proliferating source of methods and standards in Canada. And he asked if there was now, in essence, 2 varieties of ephedrine, 2 kinds of thyroid. And I think Woods was right to see drugs this way, as congealed regulatory practices, and arguing that there were likewise multiple estrogens being enacted in Canada, in part as a result of these regulatory techniques. And what else was born of this? I think there were 2 lasting effects, really, on regulation and law in Canada, and I'll just flag 2 before I close. The first was a decades-long effect on the regulation of estrogenic drugs. So in 1949, the Food and Drug Regulations are amended again. There's a continued and, in fact, increased delegation of both dosing and potency methods to industry. And the effect is to basically smooth market entry for these drugs, substances, products. And importantly, the effect of this is to continue to delegate to doctors and to patients responsibility for deciding on dosage with this array of doses to choose from. And the second effect is this use of labeling to standardize and materialize estrogenic substances. While it didn't travel to a lot of other drug contexts, with a few little exceptions, it immediately traveled to the context of cosmetic regulations. The first cosmetic regulation in Canada ever made was only for estrogenic cosmetics. And you really see the birth of Canada's approach to regulating cosmetics as something that arose out of estrogen.